Hello, I'm Les Rescio. I'm the Chief Scientist at ScitoVation. Today I'll be talking about our computational genetic toxicology screening program, which is aimed at accelerating pipelines by early identification of genotoxic liabilities. This depicts our next generation genotoxicity assessments that begin with computational tools, computational toxicology, using QSAR to identify structural alerts that are indicative of bacterial mutagenicity testing. These are highly predictive of bacterial mutagenicity, or the Ames test. Secondly, we are offering mammalian cell genotoxicity assessments using the micronucleus assay in TK6 cells and in metabolically competent HEPA-RG cells. In combining with this two traditional endpoints, we also are offering toxicogenomic integration of the TGX DDI biomarker, and these can be done separately or combined to increase predictability for assessing genotoxicity in a mammalian cell. Finally, we also have a capability at ScitoVation to do in vitro to in vivo extrapolation modeling, and this is being now used in genetic toxicology to use those data sets to identify points of departure, margin of safety that can be useful for risk assessments. This is one of the reasons why we're developing a screening program, because the genetic toxicology GLP compliant test battery can cost up to $150,000. Even a simple Ames test, a GLP compliant Ames test, which includes a rangefinder, a definitive study, and key dose formulation to meet GLP testing requirements, can cost up to $32,000. Therefore, in a positive response in any of this test will stop further development. So we need to be careful when you go into this bioassays GLP compliant test battery, which are rigid, OEC defined, and cannot be changed. You want to go into these assays with a good data set to indicate that you'll have a negative response in your drug development program, or your pesticide development can go forward, because these can definitely be a showstopper on a study. Computational tools have evolved tremendously. This is a picture done by John Ashby in 1989. This is a structure that he developed to identify structural alerts that may lead to genotoxicity. Nowadays, there's a whole number of programs that have been developed, multi-case, InStem, LeadScope, LASA, the OEC toolbox, which all can be used to identify structural alerts that may indicate genotoxicity or mutagenicity, particularly in the Ames test. But these computational tools need to conduct both what's referred to as a statistical ruling and an expert ruling. And with these two rulings, and sometimes by combining some of these software packages, you can get high accuracy, up to 95% accuracy, predicting outcomes in the Ames test. The mammalian cell lines we use are human-based cell lines, primarily because the rodent cell lines used in genetic toxicology test battery are p53 deficient and have an impaired DNA damage cellular response, and these rodent cell lines are associated with a high false positive rate due to either being a rodent cell line, inbred cell line, and they're being p53 deficient. Secondly, in the past, there's been issue with not counting enough cells using microscopy-based methods. This is overcome by using flow cytometry. And finally, many of these cell lines are used in many high-throughput screening assays like lack metabolic activation. And this is one, the cytochrome P450 in S9 is the only biotransformation system available with S9. So you have bioactivation without detoxification, and we felt that HEPA-RG cells that have both phase 1 and phase 2 metabolism are an appropriate cell line to develop as a genotoxicity assay for screening. So our assays are being done. The assays we are using are the human cell flow cytometry micronucleus assays in two 53 proficient cell lines, the human TK6 cells and HEPA-RG cells. The HEPA-RG cells, as I mentioned, are phase 1 and phase 2 metabolism compliant. They're competent, and they are compliant with OECD 487. And they are being considered as a new approach methodology to reduce reliance on animals. TGX DDI biomarker has been pioneered by Kerouac, Alfornis, and a number of others. So my group was involved as well in the early days using this particular approach. It is very accurate. We have Kerouac and her colleagues, myself, have tested approximately 100 agents. It is very accurate at identifying high specificity and high sensitivity DNA damaging agents. We can combine this with the micronucleus assay to have very high predictivity. This biomarker, the TGF-CDI biomarker, has gone through the FDA biomarker validation steps shown on the bottom here, and the current status is it's in the finals of regulatory acceptance. They're in the midst of conducting a ring trial, and it'll be able to be a FDA validated biomarker in the very near future. So finally, ScitoVation has a long history of being very active in doing physiologically based pharmacokinetic modeling, and more recently in vitro to in vivo extrapolation modeling using gene expression profiling data. And that's depicted in this slide. So we can take those gene expression data, the TGF-CDI biomarker, for instance, do biological dosimetry, identify, do reverse dosimetry, and identify concentrations that may be safe for human exposure and lead to very little risk in humans using extrapolation modeling from our human cell-based assays to in vivo. So that's our genetic toxicology testing program. This cartoon or this illustration was developed to indicate that the way we've always done it this way is deadly to science, and I think in genetic toxicology, before you go into GLP studies using these old ways to be able to do these, are what I refer to as archaic technology, i.e. the microscope, you really need to have your ducks in a row so that when you go to this, you're very certain you'll have a good outcome from and will not have any problems developing your pipeline going forward. And we'd love to be able to help you out by using our technology that is highly predictive of what might happen in a regulatory setting. And thank you very much for your attention.